Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. This episode is a special one because it's part of a series of interviews dedicated to celebrate Dr. Mark Caron, a giant and visionary in the GPCR field and beyond. In this episode, we talked to a panel of 11 guests who worked with Dr. Caron in some shape or form since the early 2000s. I also had the honor of working with Dr. Kathleen Caron, the daughter of Mark Caron, who is my co-host in this series of episodes. Before we dive into this discussion, we are thrilled to announce that our ecosystem is expanding and are thrilled to count Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology as our ecosystem partners in 2023. Become an ecosystem member yourself and join our partners and your colleagues today. The ecosystem is your GPCR-focused virtual playground. Join over 700 of your peers who had already started exploring, connecting, and collaborating better. You can explore the ecosystem by signing up and getting a free site membership. When ready, you can also get a premium membership to unlock the ecosystem's full benefits. If you'd like to register your team or company, or if you live in a developing country, please reach out to us by email at hello@drgpcr.com and we'll be happy to help you join us. The list of benefits in the ecosystem is quite long. Today, we want to highlight that as a premium member, you can get in touch directly with your peers via chat in the ecosystem, discuss GPCRs in the forum, and even ask topic-specific questions in a dedicated group that you two can create directly in the ecosystem. Are you looking for your next career opportunity? Our job board is a GPCR-focused one where you can explore different opportunities, and if you're looking to hire, you can submit your own job description. Wondering what GPCR meeting to attend next? Check out the events page where we have curated the next GPCR meetings for you. In case you're organizing a meeting, fill out the event submission form and advertise your event in the ecosystem. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Explore the possibilities by navigating the site using the direct links in the footer. Check it out today at drgpcr.com/ecosystem. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today we are recording a very special episode. This is episode 102 of the Dr. GPCR podcast, and it is part of our tribute to Dr. Mark Caron, who passed away earlier this year. Or should I say earlier last year, because when this episode airs, we're going to be in 2023. I'd like to thank you all for being here. Thank our guests for being here. And we're going to start out with a quick introduction from my co-host, Kathleen Caron. Kathleen, thank you so much for being my co-host today. So why don't we start off with a quick introduction as to who you are, and you can pass on the baton.
Yes, well, first and foremost, Yamina, thank you so much for all of your efforts in doing this. It's a wonderful, heartfelt tribute, and on behalf of myself and my sister Melissa and my brother Nelson, whose birthday is today, we'd just like to thank you and all of you who are joining us, for paying this tribute to Dad. It means the world to us. So I'll kick off the introductions, and so we're going to mention who we are and where we are, and then when we first met Mark. So I'm Kathleen Caron. I'm Mark's eldest daughter, and I'm currently a professor and chair of the Cell Biology and Physiology Department at the University of North Carolina at Chapel Hill. It's a large basic science department just down the street from Duke. I first met Mark on July 17th, 1970, when I was born, and really loved being his daughter. And so I'm going to go around what I see on my Zoom screen and just call on the next person. So the next person that I can see is Bill Wetzel.
Yeah, so I'm Bill Wetzel. I'm at Duke University. I'm in the Department of Psychiatry and Behavioral Sciences, and I also run the Mouse Behavioral Phenotyping Core. Unfortunately, I was never a student of Mark, nor was I a postdoc, but he and I were very close colleagues, and I can say that I learned a great deal from Mark, and he was my best friend at Duke.
Thank you,
So, Bill, can you pick the next person, Bill?
Oh, sure.
Yeah.
So how about Vanya?
I'm sorry. I'm Vanya Prado. I'm a professor at Western University in London, Ontario. So when I met Mark, I think it was 1999, and he was in Brazil. He came to visit, and um, I remember vividly, we were in a Brazilian restaurant having a nice food that he really enjoyed. And then we were talking about research, and he said, well, you should come to our lab. Was me, him, and Marco that he's going to talk soon. And then he said, well, you should come to our lab, and then we could make a transgenic, genetically modified animal, and would be wonderful. So Mark was a great friend, and like actually, it was family, and not only him, but everybody, because he took us to his home, and then we used to be with him, Pauline, and the kids and the grandkids, and then it was fantastic. So we a lot.
Thank you, Vanya. Please pick your next person to pass the baton to.
Let's go to Amy.
Hi, I'm Amy Ramsey, and I'm at the University of Toronto. I first met Mark in 1998 when I was still a graduate student, and we came to Mark to help us characterize a new mouse line that we had made. And working with Mark, we saw that this mouse could be useful as a model for schizophrenia, which we had never considered. So that was the beginning of a long mentorship. And I was in his lab from 1999 until 2009 as a postdoctoral fellow. So I'm going to pick Marco.
Oh, Amy, thank you. I'm Marco Prado, and as Vanya mentioned, we're both professors at the University of Western Ontario and in the Department of Physiology and Anatomy and Cell Biology. We also follow through on a lot of what we learned by years of spending time with Mark, and we run a really large core for mouse work here at Western. So Vanya's right. We first met Mark in 1999 when we were still in Brazil. And we were invited him to come for a talk, and he came several times for talks in Brazil. There's some stories behind it, because in a couple of times he forgot that he needed to get a visa to go to Brazil, which involved me to get contacts in the Foreign Affairs Ministry to go to his house and get him the visa so he could make to Brazil in time. And I remember very well, not just the first time, but all the times that we spent together in trips and in meetings, and it's been such an influence in our lives. And in fact, we are just in Canada because Mark came to our labs in Brazil and looked at our labs and said, really, you can't stay here. You got to move somewhere else. That's not going to work for you guys. And that triggered us moving to Canada. I'll tell more stories later.
Thank you, Marco.
Next. Maybe I can pick Kafui.
Hey, how you doing? I'm Kapu Drasa. I'm on faculty at Duke and an investigator at the Howard Hughes Medical Institute. I first met Mark in 2003 when I started in graduate school in the Department of Neurobiology. And I never actually had the pleasure of officially being Mark's student. I just went to his lab meetings and then wouldn't leave. And so somehow he eventually adopted me into the family. I will call on Lauren.
I'm Lauren Slosky. I'm a new assistant professor in the Department of Pharmacology at the University of Minnesota. And I first met Mark in 2015 when I came for a postdoc interview. It was the first and only postdoc interview I did. He offered me the position at the end of the day. I accepted, and he and Larry were anxious to get someone in. And so we negotiated a 60-day period in which I would write up and defend a dissertation. And by the end of 2015, I was there. And I had the honor really of being a postdoc in that group for almost exactly 6 years. I just left in the fall of 2021. And next up, can we go with Laura?
Hi, yes, I'm Laura Bond. I'm a professor and chair of molecular medicine at Scripps, now University of Florida Scripps Research Institute. I met Mark also very much like Lauren. The first time I ever met him was at my postdoc interview, my one and only postdoc interview. And it was funny because I remember, I didn't really know how to gauge it, how it was going. And by the last day, he and Pauline took me to ice cream, and he said, "So are you coming?" I said, "Well, are you offering?" And so that was pretty much our negotiation over ice cream. So I was very happy to be in the lab from 1999 to 2003.
Thank you, Laura. Can you call on someone else?
Yes, I have to do that. So I'll call on Henrik.
Hi, everyone. I'm Henrik Dolman. I'm a professor and chair in the Department of Pharmacology at the University of North Carolina, so I'm a colleague of Kathleen's. I think I've known Mark longer than I think anybody in this group, except for Kathleen, and just barely. I joined the lab. I joined Bob Lefkowitz's lab as a graduate student in 1982, and I stayed there. I stayed on for a year as a postdoc, so I was there from 1982 to 89. And Mark was at that point transitioning from, he had been a postdoc with Bob and was transitioning to run his own lab. And Mark was very much at the bench and very close to the science. And he played a very important role in my development as a scientist. We published 14 papers together, some of them good. And I'm very eternally grateful to Mark for what he has done for me personally and professionally. Let's see. Ali, have you gone?
I haven't, so I'll go. So, hi, everyone. So my name is Ali Salapur. I'm also at the University of Toronto in the Department of Pharmacology and Toxicology. So my pedigree is actually GPCRs. So I knew about Mark's work coming through grad school. And the first time I saw him was he gave a talk at the University of Montreal, which is where I did my PhD in 97. And then I periodically saw him at the GPCR retreats. And he gave a keynote speak at a GPCR retreat. And that's what prompted me to apply to do a postdoc with him. So I was in Mark's lab from 2002, end of 2002 till 2009. And, it was tremendous for me because I was coming from a GPCR background, which is one facet of Mark's lab. And a lot of people in the GPCR field only knew about the GPCR stuff. And then I came and I learned about the neurobiology of the dopamine system and dopamine neurotransmission and learned about animals and mouse behavior. So that was a great training environment for me. It's something that we have carried. And, a lot of people on this call are colleagues, mentors, people that, we look up to. So it was amazing years with Mark for sure. And I have also some other stories to tell, but looking forward to this conversation. So I'm going to ask Yasushi, with whom we overlap in Mark's lab, to go next, please.
Yeah, thank you, Ali. My name is Yasushi Masuda, calling from Japan, Far East. Thank you for Yamina for giving me this opportunity to join this podcast. I first met Mark in 2003 at Raleigh-Durham International Airport, which when is the Christmas Eve. This was written by Kathleen in the obituary in Neuron, who canceled the Christmas Eve party of his families for the first time and never happened later. So I haven't known the importance of the Christmas Eve in the US. So I just get the ticket only in the Christmas Eve to the Lauderdale International Airport. Mark and Melissa met my family and me and hugged me and welcome to US. And then Melissa and Caron and Mark took me the weekly apartment, which Mark had. Yeah, Mark has done very much, many things for me and my family. Actually, I have 3 kids, 3 daughters, who Mark cared my kids very well, to get the US very well. Yeah, actually, I first saw the name of the mark in 1986 or something, to see his name next to the Bob's name. So I have been very interested in Mark, why he was there for a long time. So this is why I chose the Mark's lab to how or who he was. So this was done by visit of 2004 as a visiting scientist. That's all. Thank you very much.
Yes, Sushi, you will be proud to know that every Christmas our family recalls your family, and we joke that we're canceling Christmas.
Yeah. So sorry. So sorry. I never imagined that.
Oh, we love it. It's wonderful memories. So I think that last on our call, I believe, is Brian. Brian.
Hello, everybody. Brian Roth, UNC Chapel Hill, Department of Pharmacology. I think the first time I ever met Mark was at a meeting in Italy in 1990. It was a serotonin meeting, and he had just cloned the first serotonin receptor, the 5-HT1A, which was initially published as an orphan. So they got 2 Nature papers out of that. They got a paper for cloning the first orphan, and then they got another paper saying that the orphan receptor is the 5-HT1A receptor. So most of my, until I moved to Chapel Hill, I didn't actually know Mark, but I would run into him at meetings, and he was, as you all know, he's a superstar, basically. And I was always, pretty much in awe of his work. I'll just give a couple of anecdotes. The first time I saw him give a talk when I was at Case Western, it was the first talk I ever saw where people showed videos, and he showed videos of endocytosis, of receptor endocytosis, which was amazing to see. And I remember about halfway through it was some named lecture there. About halfway through the lecture, he took off his coat because he said he was getting overheated and overexcited, and and then he switched into what we referred to as turbo Caron mode, where he went through 50 slides in 20 minutes. But it was like amazing. And I had the chance to meet with him just briefly. He came around to visit young assistant professors, and the advice he gave me was to publish in better journals. And I didn't didn't really know what he was talking about, because I thought molecular pharmacology and JBC were fine, always the journals I aspired to publish in. And it wasn't until many years later that I finally figured out what he was saying. But that's sort of my early remembrances of him.
Thank you, Brian. So let's jump in. Ellie, you had mentioned that we all know, and I know where you come from your PhD lab, and you were one of my teachers at the time while you were in Michelle's lab, and that's what got me into the field. But you mentioned that Mark not only contributed to the GPCR field, but to the dopamine field. So my first question, and Ellie, I'm pointing at you, but the question is for everyone. How do you think Mark influenced the GPCR field or the scientific field in general? And please feel free to jump in, but we'll start with Ellie.
So what I would say is holistic science. That's the best way that I can say this. So I knew all the arrestin, endocytosis, ERK pathways, all of that work. I knew it. I read that literature, but I didn't read the dopamine transporter transmission and I didn't read that. And then at this GPCR retreat, Mark gave a keynote last minute because one of the keynotes had canceled. And so they were looking at the meeting. They're like, who's going to replace? Okay, let's ask Mark. And luckily he had slides in his suitcase, because that's what he used to do. He would travel with his slides. So he gave a talk, and it was a science paper in '99 from Raoul's work, from a colleague of ours, on how the dopamine transporter, when you give them psychostimulants, they calm down. And I was sitting there, I was like, wow, they're giving drugs that are actually used in humans. They're getting an effect in these animals. It was a whole new world for me. And I had done, 5-6 years with Michel Bouvier, who was my PhD mentor. I wanted to do something else. And when I was in Mark's lab, and we had, of course, data clubs with, the LEFCO lab and other labs. The amazing thing to me was how the science was holistic. And Mark, maybe he told Brian to publish in better journals, but when we were trainees in the lab, he would say, just do good science. Just go do science and people will read it and cite it. Of course, we all, push to get publications in top journals, but what was the hardest for me as a scientist, and Amy and I moved together. So, many people know this, but we have moved together, but it was hard to start our own lab and not be able to do this holistic science with experts in everything. And if there was no local expertise, Mark would seek it out, right? So, we have colleagues here on the call from other universities, partially because Mark would be like, okay, we need to do this. Okay, let's call Brian, for example. Or this can be done by the Wetzel lab or other people. I have collaborators now that started in Mark's lab, former trainees, Sarah Jones, Gary Miller. I didn't know these guys because they were before me, but now they're friends and colleagues thanks to Mark, because that's where the science was going and that's what we needed to do. So that's my perception of how Mark did science. Amazing, holistic, full science from A to Z, which is not easy to do, even in 2022. So that's my thought.
Kathleen, you had an interesting comment in the first episode. I think it's worth repeating what you had said.
And I think Ali started to touch upon that. I think for me, separating myself as a scientist and just looking at what Dad did, right? This is what he did, right? It's the people on this call, and it's the hundreds of trainees, and the influence on a personal level of the success of scientists. So many lives have been touched, and so much, positive support of scientists all over the globe. And so to me, it's not about a receptor that was cloned or purified or a transporter or beta arrestin. Those are molecules, and those are great. But I know that Dad would say the most important thing are the people that he's supported. And so, that, to me, that's his biggest influence on the field, the long-lasting influence of training many, many generations of wonderful scientists.
Thank you for that.
Yeah,
Anyone else?
I'll say, unlike Ali, I never left. I wasn't kidding. I sort of showed up at lab meeting in 2003 and then like literally never left. So I finished my MD PhD and then Mark became my official mentor in my transition to faculty. And he's like, you need more mentors. So he called Brian and told me to go hang out with Brian, every week at his lab meeting too. And then when he was finally like, I'm not sure you need a faculty mentor anymore, he like started mentoring my students, right? So we had like a joint grad student and he's on thesis committees. And so for me, like, capturing this idea of his investment in people, he was always like, let's do some cool stuff together, right? And bring all of yourself into that, bring all of everything you come in with, and let's do some cool stuff together. And certainly the stuff we were doing in 2003 evolved to the stuff, we were doing later, but it was always like the experience of just doing cool stuff together.
Lauren, I see you saying yes. Maybe you want to chime in.
Say Laura or Lauren?
Lauren, but anyone can chime in. Please feel free to do so. Everyone is very, I know you're taking turns. You don't have to. Just turn on your camera, your mic, and chime in.
I was just going to wholeheartedly agree both what was said about this holistic science and also about, the major product being this community of people that wouldn't be together if not for Mark. And just having gone through or in the process of going through this experience of starting a lab, I appreciate more than ever how difficult it is to do these different independent lines of investigation where you're trying to do signal transduction in cells and behavioral pharmacology in animals and run a genetically modified mouse colony. It's just so impressive. And the fact that he was able to prioritize people and ask everyone to just show up and bring your whole self and bring your ideas. I appreciate every day what a phenomenal training experience that was. And yeah.
So I'm gonna not just agree with this, but also chime in and thinking that probably his biggest influence is not just that trained a whole bunch of us or collaborated with a whole bunch of us, but he also set an example that is really hard to follow for all of us. And I think to me, that's one of the biggest contribution, whatever meeting he was, and I had to be with him in meetings in Brazil with people that were not doing science that got published in Nature or Neuron or Cell, but he would give the time of the day for anybody that was there to tell him their story. So he was a listener. I actually, even after we moved to Canada, I used to call him every month and chat with him for 20 minutes and just kind of, check in and learn what was going on and, tell him what was going on with us as well. So that example of kindness, of being a gentleman and the more pure way of the word, really, being this person that is always there for everybody. I think that's the biggest influence that I see that he has had in science, because, whoever thinks of themselves as a good scientist cannot think of themselves as a good scientist if they don't really try to follow that example. So that to me is one of the major points of having interacted with Mark over the years.
So I sort of wanted to follow up on that. And so I would go up and talk to Mark and we'd discuss experiments. And so then he would think of one of you. And so he'd say, I've got to call Amy, or I've got to call Ali, or I've got to call Marco, and I've got to discuss an experiment, or, I need some help on something, so I'm going to call them. And so he was always really supportive of all of the people that he had, and he followed up with everybody. He was also a team player here at Duke. He always volunteered for different types of committees and different types of things. And he was always concerned with trying to help students actually in other labs, as Koff has indicated. He was a big proponent of supporting people. He supported me and several times by telling me to call a couple of pharmaceutical companies. So then I was able then to be able to get different types of grants from them and actually work on some very interesting compounds. In addition, every afternoon or every morning, he would also call his wife, Pauline, and they would sit there and chat together, in French. And, they would converse for quite some time. And he would always be, catching up with each other. And a final thing, I'll sort of tell a little story is that Mark was not the most organized person in the world. And so I would go to a meeting. And so I'd be at the meeting and, checked into my hotel and I'd run into Mark. And Mark would say, "Hey, Bill, can I stay with you? I don't have a room." And so, usually I would have already prepared for this so that I would have gotten twin beds or something like that. But he would just show up and and then we'd have a room together. And so one time I actually he decided to plan ahead. And so, Mark always looked for the cheapest place to stay. And so I found this inexpensive hotel. It was in Washington, D.C., and it looked really good. And it was not very far from the convention center. And so I booked, a room for him and a room for me. So we showed up, and it ended up that the building was under construction. And so it's you'd walk down the hall, and it was you could see the 2x4s, and there was no carpet, and you had to go down to the main floor actually to use the internet. And so we were sitting down there together using the internet. But I mean, that's sort of the way Mark was, and sort of the way in which you would travel with him. It was always an experience. And one final thing I want to say about the compassion of Mark was that he was at a scientific meeting, and one of the speakers collapsed. And so Mark went with him to the hospital, and he stayed there with the person for a couple of days to make sure that he would be okay. He rearranged his flights and everything. And so he came back after a few days from that. And so I just wanted to leave you with, some of these non-scientific type of memories that I experienced with him. I have many scientific memories also, but I think these were very special. Thank you.
Thank you for sharing, Bill. Anyone else wants to chime in?
Well, I love the story about the hotel because I remember some of those. I think we were at the same meeting. I remember this happening. And he was like this with flights too. He would make me so nervous because I am somebody who gets to the airport with plenty of time, and I like to be all checked in. And we'd be sitting in the office talking, and I'd be, "Mark, do you have a flight?" "Oh yeah, in 45 minutes." "Mark, go." So always kind of cutting it real close there.
Yeah, he traveled so much. At one point he had an executive assistant, and it was kind of a joke between my mother and I, to call her first to figure out if he was on a plane or not. It was kind of 50% shot whether he'd be in the air or on the ground. And another, I think, charming thing, if you traveled with him, this is true. He knew all of the people at the airport by first name, right? So the staff members at the American Airlines desk, they know him, and I know this because I once traveled and I had my passport, and they opened my passport. "Oh, you're Mark's daughter. Hi." So, it's really remarkable how much travel he did in his career. And he loved it. We all complain about delayed flights and lost luggage, but I think he loved it because he was really connecting the world of science, and nothing he loved more than doing that.
Yeah. And he loved to travel. Just going to say, he loved to travel just with a carry-on, as most of us do, right? But one of the memories that when he went the first time in Brazil, and he discovered that there was a lot of soapstone sculptures there in Brazil, close to where we lived. And, as you all know, he loved those. And I swear to God, I cannot understand up to this day how he managed to put so many soapstones, sculptures, and pens and everything on that small suitcase. I could not believe that he was taking. I'm sure that he dropped all his coats behind and just took the soapstones with him.
Around travel, I wanted to a quick little anecdote. I mean, it is remarkable that he came to a meeting and he had slides to fill in for a speaker, right? That already tells you how he's traveling. The other story that he told us is that he always traveled with his passport, right? So he never took a chance. And he was somewhere in the Northeast, and there was some issue with flying down to RDU from Northeast because of some storm. He went to the guys and said, route me through Toronto, Canada, because there isn't direct flight between Toronto and RDU. And the guys are like, yeah, but you need a passport to go there. He's like, aha, I got my passport. So he actually did a tour around through Toronto to get home because he had his passport, right? So this is an individual who's ready to travel, right? Always like a carry-on, passport, knows where he's going, and probably slides, just in case he has to drop and give a talk, So it is a sweet memories to cherish for sure,
Yeah, and going back to the holistic science, right? When we were talking 1999, so because we were not on the dopaminergic field, but on the cholinergic field, and then if we have if we knock out any of the important genes, the animal would not survive. He said, well, why don't you think about doing a conditional knockout? So that was quite new, and then I was just starting. I said, well, no, it's not that hard. We can start doing it. And then, so that's how we started talking about that. And then we started working on it. And then the thing is, in 2003, when we went to the lab, we went to finalize the animal, and then we figured out that there was something wrong with the mouse. The mouse was still okay, and we've been using the mouse up to today, but it was not what we were planning. So it was not going to be a conditional knockout, it was going to be a knockdown. And he said, well, so just let's fix it, let's make another one. So, that was Mark, because he wanted to go to the end, right? So for him, and it doesn't matter, it's cholinergic, it's like receptors, anything. He wanted to go to the end, and he was always to help. It was fantastic.
Yeah, I was thinking about, again, I think that what he gave us, because he had so many trainees, was he gave science this approach of, thinking about science where you're not afraid to go to a new model if you need to go to a new model. I mean, he decided to start doing zebrafish, which it's really hard to go to a new model once you're really good at one, to switch. I also think about how. So I think he trained a lot of people with a good way of approaching science. I think that makes a huge impact, and I think about how he really put the science first and ego second. It was really not about what kind of paper it was going to be. It was just really, I think he had a good nose for what was a good story. He let people pursue things, so I think he gave a lot of room to move into other fields. He wasn't afraid if that took him into an area where he wasn't necessarily the expert. And I mean, I love that he was so humble. He really was very humble, and about all of his accomplishments. And I think he taught me that the science comes first, and it really doesn't matter, and nothing else really matters. It's are you doing the science? Are you doing the science that you want to do? Answering the questions that you want to answer in the right way. So I really thank him for that. It's good to remember in a very competitive profession, to take your ego out of it.
So in being humble, it just reminded me a story. One of the people that collaborated with Mark was Rui Costa, who is now the director of the Allen Brain Institute, and that somewhat started when I sort of visited the lab that he was, and talked a little bit, and I was talking to Miguel Nicoleles at the time. I said, it's interesting, you guys are doing all these recordings, and Mark next door has all these mice, and then Rui came to me afterwards. I said, this was great because I've been telling Miguel all the time about this, and we got to see Mark, and we got to see Mark the next day. And it's kind of weird because I think Rui was renting a house or an apartment for Mark. And one of the stories that he told me is that, they were a bit lazy with cutting the grass. And he then says, and then one day I wake up and Mark Caron is mowing my lawn. That's the thing, right? Not just, because we can't forget that one thing that Mark's loved was to work, really to do the hard work, either raising cows or, I always talk to him and it's like, how many trees did you remove this month? And so that's a story that you said about humble, and here's one of the most famous scientists in a lifetime mowing your lawn because he didn't do it.
I think, Marco, that segues us to maybe a next prompt question. And so I'm going to go ahead and answer for my answer. But the question is to name a top quality, an adjective that you feel, made Mark successful. And I know that you all have been touching on this, but the one that I choose, Marco, is the work ethic, and as his children, you can imagine what it was like on the weekends, doing yard work. And so, you've seen this, and sometimes we were amazed and in wonderment of how it might have worked in the laboratory, because, Dad was always the one raking, blowing, chopping, whatever. He was a true lumberjack, absolutely.
Yeah.
Grew up on a lumber farm in northern Quebec, a maple syrup farm. And so, our weekends were picking up sticks and watching him, do it the right way, right? So he would teach us how to work the right way, and which meant watching him, sweat through lots of headbands and things. And but this work ethic, is something that is, I think, when done positively and with pride, is something that is very contagious, and so you just kind of see the joy in the hard work, and you think, wow, could I have that? Could I have that level of work ethic and drive and passion? And so, from a family perspective and also a scientist perspective, I think one of his greatest qualities was this remarkable work ethic that he had. So again, the prompt is a quality, a quality that he had that made him successful. Yeah. So
this is Brian. I would say the one quality for me is he was optimistic.
Second.
What? Yeah. I mean, he, no matter how things were going, so we had a big program project together. And he was always optimistic, basically, despite, I would say, everything. And sort of related to this, I just want to give a little anecdote. So he and I were on this editorial board together that we would meet once a week. Kathleen was on this as well, Journal of Clinical Investigation. So we would meet every week. We had a physical meeting to discuss papers. And Mark was unusual in that he always tried to find something positive about the papers. And whereas I would always try to find something negative. So we would sort of balance each other out. But he was always, if he decided to discuss a paper, he was optimistic about it, and he would defend it pretty vigorously, actually. And I think 99 times out of 100, we would all defer to him. And sort of related to that, occasionally there would be things that we would review that would be somewhat controversial. And I tended to shy away from those sorts of things that were, controversial in the GPCR field. But Mark always used to say that, and I don't know if he shared this with his trainees at all, but he always felt that an experiment was always true. Basically, if you did the experiment right, it was true. And you needed to trust those results. And he also felt that there was, if there was something out there in the literature, that it couldn't be completely wrong, basically, that there had to be some kernel of truth to that thing. And he would try to focus on, if it was true, what would be the implications of that? And that was pretty liberating to me, because I was always looking, what's wrong with this paper? Let's, take care of this as soon as possible. Very unusual. I think Kathleen would probably second that. I never saw him get upset about a paper, never raised his voice. When he would disagree, he wouldn't be disagreeable, basically. Just a wonderful, wonderful person to work with.
Yeah, if you were the author and had him as your handling editor, you were in luck.
Yes, you were in luck. If you had me, you were not in luck.
Any other?
So I find it hard to find one word to describe Mark. I like to suggest merging two words for me. Is generosity and kindness. If we can find a word that puts those two together, it would be, to me, what is really, a loving person to interact with.
Yeah, I agree with what's been said. I also would offer relatable, approachable, and I think that his stature and all that his work was, and what a star in the field, and yet, as you've said, anyone could have his ear. He would talk with anyone. He would discuss, and yeah.
About the work ethic, I just wanted to say that Mark also led by example. Every day in the lab, 8:39. Every day leaving, I'm going to say 6:30. There's a debate around the time, but he would go home for dinner every day. And he would be there on Saturday. And Laura knows the term, she put it, and we all know the term. But what I would say is that there would be times where the trainees in the lab, we would, start coming at like 9:15, 9:30, 9:45. And then every now and again, he would say, guys, you got to be here. You got to do the, So he had some expectation for the work ethic, and he led by example. This is important. Never, mean-spirited, but setting some boundaries and expectations. And the other thing that such a scientist is like, and he would say, "You're here for yourself. Remember that, right? It's for the science and for your own future, right?
It's your life."
So, yeah. So. And then the whole, generosity and all of that stuff. I do believe, again, my holistic comment about science is, I came from a lab where we had a technique, the bread technique, right? And, I came to Mark and I said, hey, we should do bread immediately. Sure, let's do this. What do you need? We need to buy a machine. Go for it. Test the machine, buy it. No inhibition, no barriers. The idea is that this is going to be good. We're going to do some good science with this. And so bread has been going on, and papers in science have been produced ever since then, right? For one of my papers, we were talking, he said, you know what, we should do electron microscopy. I'm like, okay, well, I'm not sure how we're going to do this. He's like, yeah, let me pick up the phone, call this person who's an electron microscopy expert, calls this person, and boom, we've got electron microscopy. That is the love of science and the discovery, right? So it was, contagious. There's for those of us who left, Kafui, not you, you stayed, so you enjoyed it. But for those of us who left, we had some kind of a withdrawal, it's hard. Lauren is going through this as a junior PI. It's not the same. I want to do all of this stuff, but it's not as easy. It's not trivial, So, and it was great that he would catalyze it. He would make it go, right? And for those that trained with him and actually did experiments with him, even better. Because if he was raking leaves and cutting wood, you can imagine, I don't know if he was cooking or not, but if he was cooking, he would probably be a great chef. So he wasn't, that's the only thing he didn't do. So he left that to someone else. Okay. But I suspect he would have been a great chef. That's my suspicion.
Well, but Pauline cooked so well that he couldn't do it, right? But on the other hand, have you seen the that he built for the kids? It was the most beautiful thing I've ever seen. Even had a second floor with a beautiful set of stairs and amazing. So everything that he did was fantastic. He was not scared of challenges on anything, like his science, his life, and the way he always treated family. So he was always working on weekends. It was not hard, not easy on the family. And poor Pauline had to be, by herself a lot of times, right? And I bet it was not easy on her and on you guys as well, right, Kathleen? But then, but then you get to see how passionate he was. And then the thing is, he was traveling, he was always talking about you. So everybody that knew him knew about you because, he would be talking about you. So I remember going looking for presents for Pauline, and because he wanted a specific thing, he said, well, I know this is going to look great on her, so I want to find it. And then we tried a lot and we couldn't find actually. But then, so even though he was not there, you guys were always with him.
Thank you for that. Anyone else wants to chime in on any qualities before we move on to the next related question?
I think I could just add, yes to the generosity, yes to being so humble, yes to the work ethic. And in case we haven't always said it, it felt like that work was being done with joy. By the time I was in the lab, the Friday end of the day, have a great weekend, see you tomorrow, was kind of a joke. We still said it every Friday, but we weren't always there Saturday morning. With operant behavior, sometimes we had to be. But he often said, I have the best job in the world. This is the best job in the world. And that really struck me, to see someone who is truly having the opportunity to live their passion was really powerful, and it was contagious.
Thank you, Lauren. Kathleen, you want to guide us into the next?
Yeah, so the next question related, but it chime in with additional stories that would be to think specifically of lessons that you might have learned from Mark that you take with you. And I know we've sort of been talking about these things, but if there's folks who haven't had the opportunity to chime in, lessons that you learned from him that you take forward in your success.
If I could just jump in, one of the lessons that I learned, and I wish I could remember the exact words that he said it when he was telling me, this is what I teach my undergraduate students when I'm teaching them about, receptors, is that once a receptor is used for one thing, nature can take that and use it for something else. And that, basically nature uses these, fundamental building blocks and then tweaks it and uses it for something else. And so that really stuck with me, because I work with the NMDA receptor, and glutamate is a kind of universal signaling molecule. And you think about, okay, this receptor, maybe it was maybe it was something else in bacteria. Before it was something in neurons. It was something in bacteria. It's something in, maybe even mitochondria. So thinking about like, oh, it's doing more than just two neurons talking to each other. I think that helps me to keep my mind open. So I think, I think that he was very open-minded, but that was what the one lesson that I think I have used in my own science the most.
Thanks, Amy. That's really great.
Can I say something about that? I think one of the things that Mark taught me was the interplay between technological innovations and conceptual breakthroughs. And you really can't have one without the other. And I think, Mark's legacy goes far beyond those who knew him in the lab. And so much of what we know conceptually about GPCRs, their sequence, their structure, their pharmacology, their function, it really has its origins. The way we study receptors today, it can be traced back directly to innovations, technological innovations that Mark pioneered, particularly early in his career. And we take these technologies for granted, but they were really groundbreaking when they happened. And these were things that Mark invented. And I'm going to mention three. Number one, if you want to measure GPCRs, today we do it by radioligand binding assays. I mean, that's routine. But that technology didn't exist until the mid-70s, and Mark invented that technology. He developed the first radioligand tritiated alpranolol to quantify adrenergic receptors. And without that, there's no way to measure these things. So that's a technological breakthrough that led to conceptual breakthroughs. Second, Mark was the first to successfully extract GPCRs out of cells using detergent. And again, it sounds trivial now, but before you can study it or before you can purify it, you have to get it in a form where you can solubilize it and where you can actually measure its function. And Mark found the one detergent in the Sigma catalog that was compatible with receptor-ligand interaction, and it was an obscure one, it's digitonin. But that's something that Mark did. And then the third one, which I think is also incredibly amazing, is affinity chromatography. Nowadays, we're used to MIC tagging proteins and flag tagging proteins, but those are variants on affinity chromatography. And the first GPCR to be purified was the beta-2 adrenergic receptor, and to get that out of cells, they're incredibly rare. You have to purify the protein 100,000-fold, and the only way you're going to do that is with affinity chromatography. And there was no MIC tagging or flag tagging in those days. And Mark managed to fuse alpranolol to cephros and allowed us for the first time to purify a GPCR in a form that was functional. So these are like three really, really important technological innovations invented by Mark that led to many of the conceptual breakthroughs that we take for granted now in the field. And I was thinking about, Brian, you said Mark told you to publish in more prestigious journals. And as an aside, those three innovations, radioligand binding, detergent solubilization, and affinity chromatography, they were published in BBRC, JBC, and JBC. So there are some pretty important papers in those less prestigious journals. But I'm not going to argue with you. It is better to publish in the top journals.
Thank you, Henrik, for that. Marco, please.
I don't know if it's better to publish in single name journals or not. But it was literally the first thing he said to me.
That was good advice.
It was good advice. It took me another decade before I had a science paper. Yeah, I think, his scientific contributions are immense. There's no other way to say it. Virtually everything that is going on in my lab today and what we plan to do in the future is inspired by what he did. And the field would not be where it is today without his tremendous work. I would say the only sad thing is that I don't think he was recognized for a lot of his contributions during his lifetime in the way he could have been. I nominated him for many things, some of which he got. I wish I could have nominated him for more major awards. But it's really been inspiring to me to see all the trainees and really the fond memories that the trainees have of being in his lab. I think if you were to ask people in my lab if they had any fond memories, it would probably be when I'm gone. They have a fond memory. I never would have mowed their lawn. But I don't celebrate holidays. So Christmas isn't a thing for me, but I can't imagine under any circumstances where my wife would allow me to go to the airport and pick some people up, pick them up and then find a place for them to stay. Usually she's like, so are they settled in? I mean, this is unbelievable. I don't know of any other mentor that has bent over backwards to make people so welcome in his lab in that way. I mean, I've never had that experience. I've never heard of it before, and truly, truly remarkable.
Thank you, Brian. Marco, without really wanting to put you on the spot, but you put a comment in the chat about a famous bottle of digitonin. Would you please elaborate on that?
Oh no, I think there was a story that I think that he had this. I think Ali may know this better than me, but he had a bottle of this detergent that he thought was really special, and nobody actually. It wasn't in the really stored. It's probably in a safe somewhere. I think he kept in a safe
in his office.
In his
office, literally.
Because it was that specific lot that he thought was quite good because of the sort of quality control they had at the time. So we heard that story.
It's like a good vintage of wine, the 1996 vintage or something. Yeah. Just to say quickly about what Brian said, we talk about the Caron family, right? So of course it's the nuclear family, Melissa, Kathleen, Nelson, but the trainees, we talk about the Caron family. And a lot of us are still in touch, very much so. And of course, the influence on Canada, and it's even bigger, right? Because of the French-Canadian connection. So there's always been like extra number of French speakers and people from France or Canada. So in Canada, there is a bunch of us from East Coast, less on the West Coast, but mostly on the East Coast of Canada. There's a concentration of the Caron family people. And it's because that's what it was. We were there and there was this sense of support, right? I got in the lab. I didn't know how to do stuff. I didn't know how to do animal work. He paired me up. We had small group meetings. People were training me. Other postdocs were training me. And I was training other postdocs because I was training in stuff and he would put people together. He's like, man, go work with this person. They'll show you how to do this. And you build relationships, it was amazing. It's for all of us, we've had different moments in our lives that have been, important and life-changing. My time in Mark Caron's lab was life-changing. That's as simple as it is,
Thank you, Ellie. It's a great segue.
There were some other additional contributions I think that should be noted. One is that he really took GPCRs to a cell biological perspective in terms of trafficking and the different proteins that were involved in downregulation and desensitization. And I think that was a really important contribution, which sometimes, shall I say, gets overshadowed. A second point is that he was really one of the first people to really go after functional selectivity in vivo. And, that was with Laura Bond's paper. And, from that actually came about then working with Brian and working with functional selectivity and developing compounds then that would be able to have different effects on behavior and on pharmacological responses. And then finally, while we have spent a lot of time talking about GPCRs, it's like his cloning and knocking out of the dopamine transporter really changed the field of dopamine neurotransmission and the things then that you could do to really be able to study it and to be able to examine functional consequences that occurred. And so I just wanted to highlight these additional areas.
So, Bill, I would say that there's no one today that does GPCR research the way Mark did.
Yeah.
He had basically a unique vision and unique resources. And there's no one in the field that can even approach what he did. It's a huge loss to the scientific community.
Okay. So I'm looking at our time and looking at a couple more prompts that we have that Yamina has prepared for us. They're really great. Thank you, Yamina. Let's pivot a little bit we've talked a little bit about influence on mentoring and junior scientists, but as junior scientists, like graduate students and undergraduate students who are considering a career in science might listen to this podcast, what should they know about Mark and his influence? What would be the gems that you would want to pass on to junior scientists? Laura?
I'll comment, because I remember when I was thinking about starting to look for my faculty position, I went to talk to Mark to say, what do you look for? What are you looking for in faculty applicants? And, and I don't have a specific technique. Like, I'm not a valuable electrophysiologist. I don't know how to do something really super special. I could do radioligand binding, so maybe that's special, right, Brian? But and he said to me, well, first thing, he handed me a sack of applications that he'd been reviewing that he had gotten for faculty applications that I could look at to get an idea of how that went. And then the other thing is, he said, it's not about the techniques, because techniques will change over time. It's about knowing the right questions and how to ask them and how to approach them to answer them. And I thought that was really good, because techniques do change over time. And as for him, techniques never stood in the way, right? He'd get on the phone and find the best to do what he needed to have done. So I thought that was really good advice. It was about being really focused and careful and asking relevant, good questions. We lost somebody.
Yeah. Who we had early.
Yeah.
Any others? What should junior scientists know about him or about what you learned from him?
I would go further on what Laura has said, because it's true about him. So, and he taught us that as well. And the important thing is not only, learn how to ask questions and how to answer them the best way, and never be afraid, right? And do your best. So, because for him, so he always was there 100%. And that's what I mean with do your best. So give yourself to it. So, I think that's the other thing that it was important for him.
Thank you. Anyone else wants to chime in?
Maybe just
you
go ahead. Go ahead, Amy.
I was just going to relay. He taught me about facing a conflict instead of running away from it. I was having a conflict with someone in the lab, and he said, I think you have a wrong idea about this person. I think they have the wrong idea about you. I want you two to go sit in an office and talk this out. And I'll pay for your lunch. You can go to lunch. I'll pay for the food. But I think that you've got a wrong idea. He's got a wrong idea about you. You just need to talk this through. So I think that, and he was right. And we had that conversation, and we resolved everything, and we had a better working relationship after that. So I think that what that lesson would be for a young person is, I guess, just that conflicts happened, and that you face them, and you don't make an enemy. You try to resolve it. And maybe that's why he had no enemies and so many friends. I don't know.
I was just going to say quickly, the questions are definitely important, but as Henrik mentioned, I'm sorry, he also did innovation, technical innovation, not being afraid of doing. I don't want to put Lauren on the spot here, but the work that Lauren published, last paper that she published from Mark's lab, is a heroic effort of self-admin in mice with, that is not easy work, but no fear, go for it. We'll do this. We'll find the resources. This is what we need to answer. Just do it. I came from a biochemistry background, which is a very reductionist approach, and it's embarrassing to say this out loud, but as an undergrad and a grad student, I was weak in physiology and more or less interested in it. But being in Mark's lab completely changed my mind. It flipped me. I love now the molecular and reductionist, but in his lab, I embraced the physiology and biology. So I learned it as a postdoc, something that I was taught in courses. I had to go back and sort of revisit and understand the importance of this. Almost needed like scientific maturity for me to embrace that. So, but this was Mark's lab, right? So we talk about this, is that it was from molecules to the animal. There are good labs in the world that are doing this right now, and it's great to see. But when I was coming through the system, there were maybe a handful of labs that were doing this. And by luck, I ended up in Mark's lab. So that opened my brain to the importance of both, because you also have this, people in each section, right? People are like, oh, I got to do physiology, forget about the molecules. And people that are like, oh, molecules only matter. But the truth is that you need them both, right? It's a continuum. So that's what I would say to trainees. Open your mind. You can love the reductionist, but don't forget about the physiology. If you're a physiologist, don't forget about the reduction. They go hand in hand,
Yeah, that goes back to the very first point about a holistic approach. Yeah, I try to do it too. I guess maybe he rubbed off a little bit on me. But yeah,
you've known him the longest, so it is only natural.
I guess so. Right?
In addition to the genetic connection, for sure, but that is only natural.
Yeah.
I think we went full circle. If anyone has anything else that they'd like to share around what they learned from Mark by watching him lead by example, or from his teachings that you think might be useful for junior scientists who want to contribute to science and to the GPCR field and even beyond, please feel free to turn on your mic.
So I was never his student or postdoc. I was more a collaborator, but I'm very intrigued with this idea of asking the right questions. And I wonder, so it's something I struggle to do still, is try to find a question that's, worth spending my time and the time of the folks in my lab on. How did he convey to you what would be a good question to ask? I mean, this is always the thing that I struggle with. Like, what is really a good question? I mean, there are things I'm interested in, but nobody else in the world is interested in. What is a good question? So how would he sort of extract that out of you, or encourage you to think about what is a good question? Because to me, that's the big challenge as a scientist, is try to find, some question that will lead to some sort of fundamental truth, hopefully.
Can I interject just quickly? Sorry, I don't want to hog the conversation. Quick comment that I would say is that Mark would come with a good question, but he would also be open and listen. This is the strength. It was a dialogue, right? So he would say, I think you should work on this. So you start working on this, you start reading, you come up, and then I would go to him and like, we should look at this too. And it's like, I'm not sure. So, convince me. And then you prompt. And then he's like, okay, wait a minute, this is good. So let's get some other people brain power. So it was definitely everybody had a project to start with, but the end was not necessarily a straight line. We would start with a project, and along the way, you're like, you know what, I think we should do this too. And he would very rarely put a break and put a wall in front of ideas and movement. He would be like, go for it. Do enough to convince me. And if not, kill it if it doesn't work. That's what I thought brought the science to the next levels. And when I was in the lab, I saw this with people constantly, because it was a postdoc heavy lab. People were trained, people were excited, people were driven. So he had good people. The other thing is that he had diverse, good people in the lab with different expertise, and he would leave them alone as long as they also did the important stuff that he wanted to do. And then he would elevate the stuff that we were doing. So I went to him. I said, somebody told me you should pursue dopamine toxicity idea. He said, this is really cool. Let's call Gary Miller. He can be your collaborator, co-mentor for this. That's how, I developed into something completely different for me. That's my comment.
Thank you.
Yeah, I seem to remember it was a lot of things happened in the process of a lab meeting, and him reflecting, talking out loud, thinking about things that he'd seen at a meeting, bringing that in. Oh, this makes me think of this, and so I think maybe all of the travel that he did, all of the people that he talked to, I think he definitely had his finger on the pulse of science. He was really, really involved. So I think maybe it was that combination of knowing everything that was going on in the field, and then maybe, as Ali said, listening to people, building, doing that kind of brainstorming. I feel like that happened at lab meetings pretty regularly. That was when I saw it.
Amy, when you say that, it makes me reflect. There were a few instances where Dad and I were at the same conference. One was in Japan at the International Endocrine Society meeting. And then there were a few little GPCR retreats where the organizers, I don't know if it was one of you, cheekily put both of us in the same session, even though I have nothing to do with neuropharmacology, and there were bets on who would give a better talk. So it was fun. but in the few conferences where I was able to observe, as a conference attendee, my father, something really struck me. And I was an assistant professor, so just learning. He would always sit in the first or second row. And, he had a notebook and he would ask questions. And this was so surprising to me, because my impression of the big famous people is that they always sit in the back, and they have their computer on or their cell phone, or they're kind of walking around and they're in the back, and off to the side, and they're walking out in the middle of the talk. But he was always in the front row. And I, that So, now I go to conferences and I sit in the front row. but I think that's to your point, Amy. Like it makes a difference, and you tell your kids, "Well, sit in the front of the classroom," right? But when you engage with the speaker and you're there and you're present and you can't focus on anything else other than science, I do think you draw, really important information and I think he was definitely doing that, and then coming back to lab with ideas. So I try to do it as well. I don't sit in the back.
Thank you so much, Kathleen. I think the way you've been talking about, how these ideas were, there was always an idea that came, and then through conversations, these ideas evolved into different projects. It seems what I hear is that this was an iterative process that went through multiple peer reviews, multiple lab reviews, and then ended up into all of these questions. But it was through that collaboration, through those discussions with everyone, and being the humble person that he was, he talked to everyone and anyone who can add in and weigh in on something they read, something they heard about. And I think that is phenomenal. And that's what led to all of these different projects and let all the trainees go into all of these different directions.
Yeah, I think Mark gave us a lot of freedom, and you were free to. The lab was very well funded, and so there weren't a lot of restrictions as, you can't buy 20 mice or whatever the case was. But he wasn't going to tell you no, but he would ask you, is that really what you want to spend your time on? And if you were smart enough to listen to that and really give it that second, like, do I really want to do that? Do I really want to know that? I think you benefited the most. But he was not one to say you can't do that, So I think he shared that responsibility with us, that we asked that question.
Thank you, Laura. I think we really went around all of the questions that I had. If there's anyone who would like to add anything to these stories, I think the bottle of digitonin and the safe in the office, I think is, I wish we could have a picture of it somewhere, Ali. Maybe at some point you had taken a picture of it. But with this, Kathleen, I want to let you say some final words before I close in tonight's session. Yep. Amy, you had your hand up.
Yeah, I think Henrik and I both want to say something. What I wanted to say was, and I'm sure, Laura, you remember this as well. We were at a meeting with Mark, and Sarah Jones was there, and we were all sitting at a table together. And he said, "Isn't this fun?" He says, "You know what? You guys should all come and do a sabbatical at the same time." And, I'm just so sad that we can't do that. Like, I really miss him, and I'm so, I miss him so much, and I regret that I could not have that, go back and have that time with you all again. It's so good to see you. It's so good to see you all. That's all I wanted to say.
I think I wanted to share. I wanted to share a little anecdote and an observation. We talked a lot about Mark's optimism and Mark's fearlessness, and I was reminded of an anecdote when I was a grad student. I woke up late. I was running late. It was lab meeting day. I was running late for lab meeting, and I knew I was running out of gas, and I decided to go for it. That was Mark's optimism talking. I ran out of gas, and I limped over to the side of Cameron Boulevard near the Duke campus, and I knew I was going to be late for sure. And out of desperation, I got out of my car and I stuck my thumb out. And who should pull over but Mark Caron? And I don't think he realized it was me at first. That was Mark being fearless. Anyway, we made it to lab. We made it to lab together, lab meeting together. The other thing, I just made an observation that many of us have been talking about Mark in the present tense, and I think that's a reflection of how his presence and his influence is still very much with us, and it's very much appreciated.
And with these words, thank you, everyone. Kathleen, maybe you want to say a couple of closing remarks. Oh, Brian, you had something too. Oh, we were just waving.
Just before Kathleen, I just want to say one last thing too. I actually want to thank Kathleen and her family for sharing Mark with us, because like Vanessa said, we know how much she took away from family time to make that environment, right? We all we're all people that have their own labs. We know how much dedication you have to have a mid-sized lab going all the time. And then imagine when you have 20, 30 people there, and most of the salaries depend on you. So I really want to express our thanks to you, Melissa and Nelson, and your families, and the kids, for sharing Mark with us over the years, over the parties, over Christmas, Thanksgivings, and that we all at one time or another participate with your family. Those are memories that we also love.
Lobster parties and Halloween parties, and
that's right.
Yeah. But we look at it that you all are our extended family, and we knew how happy Dad was with you all, and and by extension, so were we, right? So it was a wonderful way to grow up. I wouldn't change a thing. So. Okay, well, on that, I just want to close by saying thank you all. I look at the caliber of scientists on this call and know how busy everyone is, and it's late in the evening before the holidays. So thank you. Thank you for your warmth and your compassion and sharing these wonderful anecdotes, funny, heartwarming, insightful, and it just means the world to me and my sister and my brother, and we really want to thank you, the three of us. Thank you so much. And Yamina, thank you for organizing all of this.
Thank you, Kathleen. Thank you, everyone, for being here tonight. I really appreciate it. And I'm going to stop recording now. Before I do so, I want to wish you happy holidays, happy New Year, and we will see you all next year. Thank you for joining us and listening to this very special Dr. GPCR podcast episode. We would like to thank our guests, my co-host, Dr. Kathleen Caron, and our Dr. GPCR team members, Attila Forrest, Ines Pinero, and Montserrat Avila Zozoya. A huge thank you to our ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem. Join us today at drgpcr.com/ecosystem. You can also subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. And until next time, stay safe.